So today I'll discuss on mutation. Genetic mutation is a change in DNA sequence of the organism, and these mutations can result from either errors in the DNA replication during the cell division or exposure to some mutagens or due to some viral infection. So mutation results when changes occur in the nucleotide sequence of DNA, and then the term mutation refers to the permanent changes in the DNA sequence. And mutations in the germ cells are transmitted to the next progeny, while the mutation in the somatic cells, they are not transmissible to the progeny, but they may cause cancer and some congenital malfunctions. Now, causes of mutations. They can be error in replication. Errors can be if some mismatch base pair is not corrected during the proofreading phenomena in post-replication repair process, or error due to the recombination events of the DNA or spontaneous changes in the DNA, like deamination of cytosine to uracil or is spontaneous depurination. Then certain environmental factors can also be the culprits, like chemical mutagens and irradiations, like UV light or ionizing radiations can alter the structure of DNA. So this is the overall classification of types of genetic mutations. So these mutations can be of two types, the base substitution or the point mutation. They can be either transition type or transversion type. Then second type are frame shift mutation. Then base substitution can be of three types. They can be silent mutation, manifesting no effect, or missense mutations or nonsense mutations. Missense can be acceptable, partially acceptable, or unacceptable. While the frame shift mutations can be either deletion or insertion type. So we will see one by one. First, we will see understand the concept of point mutation. As the name suggests, in this particular type of mutation, only one base in the DNA is altered. Okay, the single base change can be of transition or transversion type. Transition means if the purine base is replaced by some purine base or pyrimidine by a pyrimidine. Like example, these are the example of transition mutation. Here, the thymine can be replaced by cytosine or vice versa. Or similarly, the purine base adenine can be replaced by guanine or vice versa. So this is the transition mutation, means in the same category of the bases. Here the transversion, in the transversion mutation, a purine base can be replaced by a pyrimidine one, like guanine. A thymine can be replaced by guanine or vice versa, or cytosine can be replaced by adenine. So this kind of cross changes of the bases, these are known as transversion mutations. Then what are the effects of these point mutation? They can be silent, they can be missense, or they can be nonsense. Silent mutation means there is no detectable effect, leads to the formation of codon synonymous or no change in the amino acid sequence of protein. Means the if suppose the codon changes from CGA to CGG by alteration of A by G. Transition, this is an example of transition mutation, but it does not affect the protein because both of these codon are specifying the arginine, right? In the mesh sense mutation, a different amino acid is incorporated at the corresponding site in the protein due to the change in genetic codon, which is responsible for entry of different amino acid. Then depending upon the location of mistaken amino acid in a specific protein, the missense mutations can be acceptable, partially acceptable, or unacceptable with respect to the function of that protein. So let's see acceptable missense mutation. For example, hemoglobin Hikari. What is this type? This hemoglobin has asparagine substituted for lysine. It is substituent for lysine at the 61 position in the beta globin chain. And then this HB Hikari is a type of transversion mutation. So what is happening here? Initially it was AA, it is changed to AAC. So it was responsible for a lysine. Now it is changing to genome, which is to a genetic code, which is responsible for attachment of arginine. So this is a transversion type, and this is known as hemoglobin Hikari. Acceptable. This is an acceptable missense mutation because it doesn't leads to a drastic change in the hemoglobin characteristic. Then partially accepted missense mutations. Example is sickle cell hemoglobin. Sickled hemoglobin, in which the normal amino acid position sixth of the beta chain, the glutamic acid now being replaced by valine, a very well-known example. So corresponding transversion might be either GGA or GAG. You can see it here. GAA is being replaced by U. Adenine is being replaced by uracil. So it is now converting to GUA. Or had it been GAG, so that is also, code for glutamic acid. It can be GUG. So that codes for valine. So, in both the condition, valine is now replacing the glutamic acid. Now, unacceptable missense mutations means if the mutation has happened, and this is leading to a type of lethal characteristics or unacceptable characteristic in the hemoglobin. So, for example, hemoglobin N, that is the methemoglobin, in which the normal amino acid in position 58 of the alpha chain, that is histidine, has been replaced by tyrosine, and the non-functional hemoglobin molecule is now generated. You can see it here. This codon is now CAU changed to UAU. C is replacing C is replaced by U. So or CAC is converting to UAC. So A is this kind of no change will this C is replacing here U. So what is happening? Histidine is being replaced by tyrosine. So this lead to the this functionless or non-functional hemoglobin, methemoglobin. So this is unacceptable. Then nonsense mutation lead to the conversion of an amino acid uh, codon to stop or the nonsense codon. You can see here in this example the UGG that is coding for tryptophan. Now it is changed to UGA. So that is equal to nonsense codon. So nonsense mutations causes the premature termination of any polypeptide chain. We can take the example of thalassemia, in which the codon 17 of the beta chain is now changed from UGG to UGA and results in conversion of codon tryptophan to a nonsense codon. So incomplete termination of polypeptide chain. So in the frame shift mutation, so this frame shift mutation occurs if there is an insertion or deletion of one or two nucleotide in the DNA, and that leads to the altered messenger RNA. So this is the normal messenger RNA, and these are the codons sequences there. UCU is here in this particular normal, which is being replaced by one U is now lost. One U is being lost. So there is a shifting of reading framework. So now it turns to AUG, GCC, and CU because this CU one U is being lost. One U is lost. This U was being lost. So now this framework is being shifted here, and this G will be this now U will be accommodated here. So it will like CUU. This U will be shifted here because this U is being lost. So how can we read the next frame? It will be like CUU and GCA, then AAG. One nucleotide is now reverting back. It's now added to the previous nucleotide codon. So this is how happening. This is the deletion mutation by one nucleotide. Entirely different protein will be coded because of one alteration of one nucleotide only, the whole frame is now changed. So all altered proteins, amino acids are added here. Okay. So in the next insertion mutation, what is happening here? Suppose there is an addition of cytosine is inserted here. So this is initially it was UCU. So what is happening? This one C is added here. So it will become CUC, and this is now shifted to, now it is shifted to another codon. So it will become like UUG, and its C will be shifted here. So whole frame will be shifted. So it will be read as CUG, UU, A, CC, and so on. So now because of this insertion, again the same phenomena happen. Altered kind of amino acids are added here. So whole frame is protein frame is also changed. So entirely different protein than the normal is being formed. So this is the frame shift mutation.